I want to say thank you so much to everybody for coming, and also thanks so much to the Neurological Foundation for giving me this opportunity, because I love talking about what I do, and it's really nice to be able to share the type of research that I'm doing with everybody here today. So my kind of passion in research is trying to understand and examine brain cell activity, so we're really looking at the function of the brain in health and in disease as well. So just to give you a little bit of an overview of what I'm going to talk about today, I'm going to start and just give a very general introduction on the brain and some of the techniques that we use to study it. And then I'm going to go into the types of technology that we use in the lab to actually use to actually image brain activity in rodents. And I'll give you an overview of the research projects that we're working on right now and some of the results that we've found. And then I'll just give a bit of a summary and summarise our current goals that we're working on as well. So the human brain, it is a very, very complex organ, and it contains a huge number of neurons, 10 billion and 10 billion trillion synapses, which are the connections which allow the brain cells to talk to each other. And you can see on this picture here that it is kind of a weird looking structure. So it has all these folds on the surface, and it has different parts as well. And actually the different parts of the brain are specialised to do different functions. So at the bottom here, this part here is called the midbrain, and that's kind of where all the basic functions are localised, so that control respiration and temperature control and eating and those types of things. Then you also have the cerebellum at the back here, which is important for movement. And then most of the brain that you can see here is really the cortex. And this is where kind of all our higher level functions take place. So at the back of the brain is where vision is processed. At the sides you have auditory parts of the brain. And then right up here at the top is where kind of all your decision making and all those kind of really difficult tasks are processed. So in those, a huge number of brain cells, they're actually all very, very well structured, and their structure is very specialised in terms of what they actually do. So this is a kind of just a basic cartoon of a neuron here, and you can see that it's got different parts, and the parts actually have different roles. So the cell body, or the soma, which is here, it's where the DNA is located. And then you also have the dendrites, which surround the cell body. And these are the parts of the brain cell that receive inputs from other brain cells. Then you have the axon, which extends out here, and this can be very long. So if we're talking about a motor neuron, this actually goes from your cortex all the way down your spinal cord to your muscles. So these axons can be incredibly long. And this is the sending part of the neuron. So it sends out the signals to other brain cells. And yeah, the connections, like I said, between brain cells are called synapses, and there's each brain cell has hundreds and hundreds of these. So this cartoon here is a bit fancier looking, but it kind of gives you an idea of how the brain is actually working functionally, so I really like it. So you can kind of see these yellow spots here, and they look a bit like sparks. And that's because the brain actually uses electrical activity as well as chemical activity to spread the signals. So when this brain cell, for example, gets enough inputs, so if it receives enough synaptic inputs, it will become active, and that is electrical activity. So it fires what is known as an action potential, and it's kind of like a spark, which then spreads down the axon, and then it will actually send that message out to other neurons. And when it does that, it releases neurotransmitters, and these are kind of the chemicals in the brain. So sometimes you'll hear about different disorders being called a chemical imbalance in the brain, and that really means that the neurotransmitters, kind of the levels, are a bit out of balance, and that can lead to various different diseases and disorders. So what does an actual brain cell really look like? So this is a photo of a brain cell which I actually took a long time ago during my PhD. And in this, so this brain cell, what I did was actually dissect up a brain and kind of mush it up and then plate those brain cells out onto a piece of glass so they grow flat in a monolayer. And then you can kind of stain them and make different parts of the cell light up, and it looks very beautiful, as you can see here. So the way that this brain cell has been stained is that the green part, which you can see, is the dendrites of that cell. And then the red spots, which hopefully you can see, there's hundreds of them there. Those are all the connections. So those are the synapses where the cell is actually getting input from other cells. So yeah, like I said, this is grown in culture, and then it's been fixed and it's been stained. And you can look at all sorts of different things by using that technique. But when you're studying brain cells, there's kind of two different approaches that you can take. You can either look at the structure, which is kind of shown on this side of the diagram, or the function, which is more like the circuit board, which you see on the other side of that diagram. So you can either look at fixed tissue or live tissue. So if we're talking about fixed tissue, this could be post-mortem tissue from a human or from an animal. So I'm sure you know that the Neurological Foundation has been massively involved in getting human samples of brains that are used to study all sorts of different things. And when you have a piece of fixed tissue, you can do lots of different things, but one example is to actually stain for different cellular components or look at how different structures have changed in size. And this is really, really important for looking at the structural changes in the brain. So if you have a control brain or a healthy brain versus a diseased brain. But the structure doesn't kind of tell you the full picture. You really want to actually have living tissue to be able to understand what's really going on. So you want to be able to actually look at the electrical activity in a live piece of brain tissue to really understand how that tissue is working. So in terms of getting live tissue, most of the work that is done in neuroscience these days is actually done in animals, but it is actually possible to do some type of electrical activity measurements in human samples. So sometimes when people are getting surgeries for epilepsy, the neurosurgeons will actually do some recordings of individual brain cells just to check that they're not taking out a piece of tissue that's actually crucially involved in some function like movement or hearing. But what this live tissue really allows us to do is to record electrical activity, so take videos of electrical activity so that we can really try and understand it. And you can also watch different cellular processes in real time, such as neurons growing in different ways and making connections with each other. So this type of live tissue is really essential if we're going to look at functional differences in the brain. And you've probably heard of some different types of kind of imaging which are done in humans. One example is magnetic resonance imaging, or MRI. And this technique is actually used to look at both the structure and the function of the brain. So here you can see a person inside a scanner. It's a really large instrument. And then you can see the kind of detail of the brain that you can see in this image. So this is kind of a slice right through the center of the head. So this technique is really cool for looking at structure, but it also can be used for looking at brain activity. So sometimes when people get put inside the scanner, they're told to do different things. Maybe they listen to music, or they're shown some different photos of different things. And then what you can actually do is record the blood flow changes that result from that kind of stimulation. So if a part of the brain becomes active, it will have a whole bunch of blood travelling to that region. And you can actually track those blood flow changes to see which areas were activated. But the thing with doing that is that its resolution is a bit limited in terms of how fast that blood flow is actually happening after the neurons are activated. And also you can't resolve what individual brain cells are doing, which is what I'm really passionate about. So that's where we come to using animals. So all of the work that I do is working with animals, so rodents, mice and rats. And these rodents are really widely used in neuroscience. And there's so many different tools that are available now to study these animals and to also genetically modify them so that we can study different types of disease. And there's lots of different tools that we have, especially for looking inside the brain and studying how brain cells actually work, and even studying them over time. And I just want to say, because I am going to show some pictures and some videos of the animals that are used in our research, that everything we do is really strictly controlled by the Animal Ethics Committee at the University of Auckland. And, you know, we don't take this animal work lightly. We really make sure that we respect the animals and we treat them with the utmost of care. And the type of work that I am doing, it's It's kind of quite novel in New Zealand, so it's been quite a process getting the approval to do it. And we've worked very closely with the university vet to actually make sure that we're treating our animals well during the surgeries and monitoring them well so that they're not in pain. Cool, but then you might ask, but what can we learn about the human brain from studying a rodent brain? Because obviously they are quite different. One of the obvious differences is that the size is much smaller. So the mouse brain down here, and to the human brain here, and some different monkey brains in between. So yes, the rodent brain is a lot smaller. And if you kind of blow the mouse brain up, so it's a similar size to a human brain, and put them side by side, then there are still some really striking differences as well. So a couple of things that you would notice is that in the mouse brain, this is the olfactory bulb at the front, which is involved in a sense of smell. It's really quite large. In humans, you can't even see it here. It's still there, but it's very small, and it's kind of tucked away underneath the cortex. Same thing with the cerebellum. In the mouse, it's very pronounced, whereas in the human, it's actually hidden kind of down at the back. But the part of the brain that I study, which is the cortex, which is here, it is different between the mouse and the human. It's very smooth in the mouse, whereas in humans, it's very sort of convoluted and folded. And that's because with evolution, the brain got more and more complex, and it had to fold to actually fit inside the skull. So these folds actually enable the brain to have the maximum amount of brain cells, but still fit inside our skulls. But one of the things that's actually quite striking if you look at the cortex of mice versus humans, so mouse here and human here, is that if you look at what functions the different parts of the brain do, it's completely overlapped, so it's very much the same. And of course, this is because we had a common ancestor. So if you look at the different sort of sensory parts of the cortex, then what you can see is that at the back here in red, that's the visual part of the mouse brain, which is also at the back of our brain. The auditory part is more lateral, so you can see here. The sensory part is more in the centre here, and then the blue part is actually the motor part, or the frontal part of the brain. And yeah, that's where we kind of differ quite a lot from mice. So you can see that this area is massively expanded in humans, and that's because we are more intelligent. We have language, we can plan, we can have very complex social interactions, and that part of the brain has grown a lot bigger in humans. But still, I hope you really appreciate that there are massive similarities between the brains of mice and humans. And actually, if you look even inside the brain at the way the cells are organised, it's very similar. So what are the kinds of experiments that we can do with rodents? So one of the things that is done quite a lot is to dissect out the brain and then slice it up and then stain for different things. So when I talk about staining, so every gene in our DNA actually is code for a protein, and we know the localization of hundreds, thousands of different proteins in the rat brain or the mouse brain, which this is just an example that you can see here. And we know a lot about what all the different regions in the rodent brain are doing and what type of proteins they express, what the functions are. And yeah, it's a really high level of structural detail that we have about the rodent brain. But I think that it's really important not to just, of course it's important to do experiments where you slice up the rat brain and look at what's going on, but it's even more important to do experiments where the brain is kept intact. And this is a really cool study that was published just recently, where they were able to actually map whole neurons in the rat brain. And just to give you kind of an idea of the scale here. So in this figure here, what you can see in pink is one brain cell, and it has these huge axons that spread all through the brain. So the scale of this is about 10 millimeters across, and this is just one cell that's made all these different connections right across the brain. And this study was really cool because that's the first time that people have actually been able to see the extent that these brain cells are spreading. It's actually quite a difficult thing to do. So they use some pretty cool techniques to do this, to image the whole brain, but then they had to actually trace where each of these processes went, and they actually had a whole team of people that did that sat behind the computers and traced where these connections went. And they actually managed to do this on 300 cells, and you can see the complexity. But of course, that's just a very small proportion of the number of cells in the brain. This gives you a really good picture of how structurally complex the brain really is, and how kind of vast the processes of different neurons go. So for the mouse brain, there's 70 million neurons. So you can imagine that if they did that for every single one, then this would just be a complete mess. But the type of research that I'm really interested in is not really looking at structure, it's more looking at function. And we have some really cool ways that we can actually look at function in the rodent brain. So this is an example here of a study where they've actually removed the skull and replaced it with a transparent window so that you can actually look inside the brain over a large period of time. So this is showing 6 weeks to 36 weeks. And here you can see that there's really a window that they could look inside the brain for this whole period of time. And there's quite a few really beautiful studies now where they do this, and they kind of look at how the brain is functioning at one time point, and then they will train the animal to do a certain task or a certain behavior, and then look at how the brain has changed and how the cellular responses have changed as that animal has learned that behavior. And it's a really, really powerful way to understand what the brain is actually doing and how it's changing when learning happens. So the technique that I use a lot is to actually look at activity of individual brain cells, and you can see a video of that here. So in the last few decades, there's just been an explosion of the sort of methods that we have to do these type of experiments. And the type of microscopes that we use are also developing quite rapidly as well. So this technique allows us to really take these videos of large populations of brain cells at the same time and see when they are active. So when they are electrically active, we see that kind of as a flash of light, as you can see in this video. And I don't know if you can see too well, but in this video, each of these little circles is actually an individual brain cell. So here, just in this recording, we're looking at hundreds of different neurons at the same time. And hopefully that gives you kind of an idea of how powerful this technique is, because you can look at what so many individual neurons are doing and try and understand what role they actually have in behavior. But, so a little bit of the caveat of this type of research is that it can be very expensive. So I worked, after my PhD, I moved overseas and I worked in Amsterdam for 5.5 years. And I had a very nice microscope there, which was basically my own to use. And it's like a multi-million-dollar machine. But when I moved back to New Zealand and I wanted to kind of continue this type of research that I'd learned, I kind of quickly became aware that the type of systems that you want are very expensive and sort of out of reach for most people, sadly. So I had to kind of think of a way to get around this problem. So I teamed up with some people in the physics department at the University of Auckland, so Frederik van Holsbeek, which is shown down here, and together with a Postdoctoral Fellow, Juliette, who's working with me, and a PhD student, Ashley. We're actually building our own customised microscope. And one of the very expensive parts of these types of microscope is the laser. They can cost anywhere between sort of a couple of $100,000 and $500,000. But we actually teamed up with some people who can build lasers themselves for a much smaller price. So what this microscope really allows us to do, I'm not going to go into details of how it works, it's called a 2-photon microscope, but what it really allows us to do is go from a situation where we can see areas of the brain lighting up to actually being able to resolve individual brain cells. So in this picture here, each of these circles is an individual brain cell, and we can actually see what each of them is doing at each moment in time that we're recording. But of course, it's a homemade microscope, so it doesn't exactly look as beautiful as a commercial system would look, but I assure you, it can work just as well. So this is actually how our laser looks here, and how the microscope looks here. Because really what a microscope is a light source, so a laser or sometimes LEDs, and then a selection of different mirrors and an objective that allow you to focus on a sample and actually capture the light from that sample. So yeah, it doesn't look the most beautiful. Well, I actually think it looks very beautiful, but it works. And so we're still in the process of really getting this up and running exactly the way we want it to be, but it's going really well, and it's been really cool working with physicists because it's not my background, but sometimes we have these conversations where we aren't exactly speaking the same language, but yeah, as time goes on, we're getting better and better at understanding each other. It was quite funny when we were searching for a PhD student for this project. I was saying to Frederic, I think we should get someone with a physics background because, yeah, then we can teach them the neuroscience part of it. That'll be easy. And she was like, no, I think we should get someone with a neuroscience background because I can teach the physics part. That'll be easy. But in the end, we went with someone with a physics background, and actually it turns out that some of the most amazing neuroscientists in the world right now are physicists. So I think that Ashley will go very far, and I'm really excited about this project. So the type of work that I really do is to get an animal, a mouse in this case, and put it underneath a microscope and then image brain activity. And sometimes we use anaesthetized animals, which in this case, this mouse, which is about 10 days old, is anaesthetized. So here you can see a little tube, and that's how we deliver a gas anesthetic, which keeps the mouse asleep while we're recording. But we are actually moving towards doing experiments in awake and behaving animals as well. So this microscope that we are building, like I said, it allows us to go from sort of the type of recording that you would see here, where you can see areas lighting up, to really being able to resolve individual neurons. So in this video here, you can actually see individual brain cells flashing. And that's the type of recording that we really want to be able to understand what each brain cell is doing. So then this is the type of data that we actually get out of a recording like this. So here is a large area of brain, and each of these circles that you can see here, they look a little bit like donuts in this case, is a brain cell. Here it looks a little bit darker, and that's because there's actually a blood vessel going across this area of the brain. But yeah, so I've put circles on a few different example cells here, and then you can see sort of the trace of activity that we get from each of those cells. So when those cells become active, we see that as an increase in the amount of light, which you can see here. So we can really look at and see, okay, well, this brain cell is active at this moment in time, whereas these ones aren't, and try and really understand what's going on at a sort of network level. So like I said before, rodents are really useful for research because you can actually model a whole bunch of different diseases and disorders and even injuries in rodents. So there's been a lot of genetic studies done in humans for years now, and these have really revealed the genetic changes that take place in disorders and diseases. So yeah, the genetic mutations or the genetic changes. And these sort of mutations that are known in humans can then be replicated in animals. And by doing that, you can actually recreate the symptoms of disease. And this really enables you to kind of understand the symptoms and examine the cellular mechanisms that are taking place in these diseases. And two of the diseases that I look at are autism spectrum disorder and also Alzheimer's disease, and also just sort of started in the last year or so looking at spinal cord injury. So this is a traumatic injury model, which we can model in rats. So I'm going to talk just a snippet about what we're doing in each of these diseases, and a little bit of background as well. So for autism spectrum disorder, it's really quite a complex disorder, and it is a spectrum. So there's really a range of severity of the different symptoms or phenotypes that people display with. So some of the common sort of things that you will hear about autism spectrum disorder are that people have trouble making eye contact, are not able to sustain a long conversation. They might have difficulty listening or communicating. They might be slow to respond to verbal requests. And also they can sometimes be sensitive to different sensory inputs. So sometimes noises can have a big effect on people, or bright lights. And there's also other sensory issues, so like sometimes sensitivity to touch. But also there can be other advantages, you could say, where people can have very good memories or are really good with numbers or facts. And in terms of autism spectrum disorder, it's a spectrum, and not everybody displays the same symptoms or signs, but the sort of sensory issues are where my interest really lies, and especially the auditory changes that can take place. So autism spectrum disorder is really complex genetically. There's been actually hundreds of genes which are associated with autism spectrum disorder. So different sort of genetic changes will lead to different symptoms. There's a really array of different kind of phenotypes that people with autism can display. But many of the mutations are actually found at the connections between brain cells, so they're synaptic proteins which are affected in autism. So the way that brain cells are talking to each other is somehow affected. But so now, because there's so many kind of mutations that have been revealed, there's actually quite a few different mouse models. But the thing a bit is, because there's so many different genes that are implicated, not each mouse model will be a perfect representation of disease. So some of them only represent about 1% of the mutations found in the human population. So it is very complex, but each mouse model kind of offers a little bit of insight into what's going on in the brain. So like I said, the sensory sort of symptoms are something that I'm really interested in, and especially hearing and auditory processing. So if we think about hearing, what happens is the sound comes in through the ear canal, and then it vibrates these bones in the inner ear, and then that sound is actually detected by the cochlea. And the cochlea is this beautiful snail-like structure that you can see here. And the cochlea and all the other parts of the auditory system actually have this really well-organised kind of way that they're structured. So the cochlea itself, as you can see on the key down here, so the lighter colour represents higher frequency sounds, and the darker colour represents lower frequency sounds. So the cochlea itself detects higher frequency sound at the beginning, and then through to lower frequency sounds in the centre. And this kind of organisation, which is known as a tone map or a tonotopic map, is present at every different part of the brain which is actually processing that auditory information. And so these here, this is quite a complex diagram. This is showing the different auditory centres which are found in the brainstem. But this information does also go all the way up to the cortex, so it's located in the lateral part of the brain. And here in this diagram, what you can see is that, so this is high frequency sounds here and low frequency sounds here, so lower tones. And you can see that this is really well organised. And our actual question that we're trying to answer is whether this is as well organised in the brain of a mouse which has autism spectrum disorder. Because our idea is that perhaps this map isn't quite so tidy or so well organised, and of course that would have implications for the way that sound is processed, and it could actually lead to difficulties with processing language and communication, which can actually then lead to antisocial behaviour. So just on this side here, you can see how this looks in a rodent brain. So the auditory cortex is actually made up of several different fields, and each have their own tonotopic map. So first of all, we started, so this kind of hearing and auditory processing question is really not very well explored in mouse models of autism spectrum disorder. So what we did, first of all, was to look into our mice and just to check their hearing to see if they're actually hearing normally. So this was work done by a master's student of mine, Minchell. And what he did was actually to record auditory brainstem responses, which can be recorded just from the scalp of a mouse. So he would deliver tones to the animals and then record the responses in the brainstem. And what he actually found was that these mice that we're working with have normal hearing when they're young adults. But that doesn't necessarily, so their hearing is normal, but that doesn't necessarily mean that the way that they process that sound is normal. So we started looking further up in the brain, in the cortex, and trying to map the different responses that you have to different tones. So what we do is we get a mouse underneath a microscope and we play tones of different volumes and different frequencies, so high versus low tones, and then we actually look at which parts of the brain light up when we play that tone. And we do this with a whole bunch of different tones, and then we can kind of map these responses in terms of what frequency they are. And this was done by a master's student in the lab, Maya. So again, just kind of going through her experiment. So she had the mouse underneath the microscope and then imaged quite a large part of brain. So this is about 3 millimeters by 3 millimeters. Played all different tones and then looked at which area responded to each different tone. And by doing that, she was able to generate these type of maps, which range from low to high frequency. And she then went on and analysed these maps in a lot more detail. And what she actually found when she compared the autism spectrum disorder mice to wild-type mice or neurotypical mice is that the area that was being activated by sound was larger in the ASD mice. So here's the wild-type mouse here. This is area, and you can see that for the ASD mice, it's a larger area that's being activated here. So the hit in red, this is these are mice that have one gene affected, and the knockout has 2 genes affected. So you can see that they kind of have an intermediate phenotype here. Another thing that she found was that the variability of sort of the areas that became active by the different tones was higher in the knockout mice. So we think that actually the sound are being processed differently in these mice. So there's a more poorly organised cortical representation of sound in ASD. And we're further expanding these studies at the moment, and my PhD student Pang Yang is actually looking into this in a lot more detail. But yeah, what we can say now is that there really does seem to be something going on here, that the sound isn't represented exactly the same in these mice, and this can really have other effects, such as, the ability to communicate and to socialise. Okay, so now I'm going to move on to a really cool technique that we've just been setting up in the last sort of year or so, and that is using miniaturized microscopes for imaging in animals which are actually running around. So these miniaturized microscopes, we call them Miniscopes. They have all the normal parts that you would see in like a full bench-sized microscope, but shrunk down to very, very teeny tiny versions. And the really cool thing about these miniaturized microscopes or miniscopes is that they only weigh 3 grams. So you can actually mount them onto an animal and it can run around and you can record its brain activity while it is behaving. And this work has really been set up by a postdoc in the lab, Juliette. So these are some photos of how that actually looks. So normally when the mouse is just living its everyday life in its home cage, it has this base plate implanted and a lens implanted which goes into its brain. And this base plate is very light, and the mice tolerate this very well. But when you actually want to record, you then clip the Miniscope on, which you can see here, and then you shine some light into the brain, and then you record the brain activity. And it does look kind of large here, but it is really, really light. So 3 grams, like I said, which is a very small percentage of the mouse's actual weight. So we'd kind of clip the Miniscope on for less than 30 minutes a day and record while the mouse is doing different behavioural tests. And I'm going to show you some videos of what this really looks like now. So this is the mouse just when it's got the base plate implanted, and you can see that it's happy, it's running around, it can move very well, and everything. So then this is now a movie of the mouse wearing the Miniscope. So you can see that it can run around, it's pretty happy, it's exploring, it's having a look around, and they actually get used to wearing the Miniscope quite rapidly. So we do a little bit of habituation with them, where you put it on for a short amount of time each day, and then they get used to it, they behave normally. And overseas there's been really extensive testing with these Miniscopes, and they don't really affect the behaviour of the mice, so you can study behaviour quite well with these. So then this is to show the type of data that we actually get from these Miniscope recordings. So the raw data looks a little bit of a mess, and you actually have some sort of movement artifacts in there because, of course, the mouse is running around, so you can have some small movement artifacts that you need to correct for. But after you've corrected for those, aligned your videos, then you can actually, it might be a little bit dim here, but you can actually see the brain cells lighting up. And what we can do then is then correlate that brain cell activity with behaviour and try and really understand what each brain cell is doing in each behaviour. So now we're just really in the process of getting data with these Miniscopes. So Hamish, who's doing his honours with us this year, and it's been a pretty difficult year for honours students in the lab, but they have actually managed to have some time in the lab and get these experiments going. So he is using these Miniscopes in this mouse model of autism spectrum disorder, which I've talked about before. And he's doing a range of different social and anxiety type tests with these mice. So one of the social tests that he's doing is where you actually, you have another mouse sort of in the arena with the mouse that's wearing the Miniscope, and you can measure how much time the mouse that you're recording spends near the other mouse. So yeah, like I say, we're just running these experiments now, but we think that the data we're going to get is going to be really exciting. So another disease that we're also looking into is Alzheimer's disease. And probably you know something about this disease. It's quite common. It's the most common form of dementia. And the most common symptom that people have is memory loss, but this actually proceeds to confusion and sort of loss of a bunch of different functions. And structurally, we know quite well what is going on. There's a massive loss of neurons in Alzheimer's disease, and also a massive loss of the connections between brain cells. So here you can see what a healthy brain looks like compared to a severe Alzheimer's disease brain, and you can see how much it's really shrunk. So there's, many brain cells have been lost here. But so even though we kind of know what's going on structurally, we don't really know how the brain cells that are still present are functioning. So there hasn't been a lot of work looking at living tissue in Alzheimer's disease. So that's why we have started to apply this Miniscope technique to actually look inside the brains of a mouse model of Alzheimer's disease while they're doing memory tests. So we know that this mouse model, they have impaired memory. And we want to try and understand how the brain cells that are surviving, how they're actually functioning. And we hope that this will reveal and help us to better understand really what's going on in the cells that are still alive and are still there. And maybe this could lead to some different treatments that could help memory. So the final project that I just want to talk about a little bit is about spinal cord injury. And so spinal cord injury is typically a traumatic injury, which leads to damage to the spinal cord. And typically this happens in motor vehicle accidents or sports and falls. And what happens like at the time of injury is that there's kind of an acute damage to the tracts in the spinal cord. But then after the injury, there's also a secondary damage that happens. And this is because you get an inflammatory response in the spinal cord, which then goes on for several weeks and it causes further damage. And it actually kind of impedes the healing that would take place normally. In addition to this, so there's a lot of changes in the spinal cord, of course, but also the circuits in the brain which connect up to the spinal cord are actually impaired and affected as well. So there's quite a big reorganization that takes place in the brain, and this can actually cause some symptoms of spinal cord injury, such as phantom pain and things like that. So, maybe you guessed already, but we really want to apply these new technologies to try and better understand what is going on functionally in a rat model of spinal cord injury. And this model has been really well established in rats. So it's known that the rats, they are injured and they lose function of their hind limbs. So they can still move, but their hind limbs are quite paralysed. But with time, this kind of starts to improve. And my collaborator, Simon O'Carroll, you can see down here, he's actually discovered a treatment which prevents some of the secondary damage that happens in spinal cord injury. So when rats are given this treatment, they actually recover their hind limb function much faster than they normally would. But we really don't understand what's then going on in the spinal cord or in the brain in these rats that are treated. So we want to apply these techniques so that we can actually understand those circuit changes that are taking place in the brain and in the spinal cord following the injury and also the treatment with this drug that Simon has discovered. And this work is done by a PhD student, Zara, you can see here. And just to kind of summarise what she is setting up and now starting to do. So we have different plans for this study. So one part of it is using the Miniscopes, so either mounted on the head of the rat so that we can kind of understand those cortical changes that are taking place and have big effects in terms of recovery. So we're planning on looking at the sensory parts of the cortex and also the motor parts of the cortex. But then we're also planning to mount the Miniscopes on the spine of the rats. And some other people have done this overseas, and we're working towards doing it ourselves at the moment. So then we can really look into the spinal cord and see how the activity in the spinal cord is changing. And we're planning to look at sensory neurons in the spinal cord, but also looking at astrocytes, because the astrocytes are actually the cells which are controlling this secondary response to this inflammation. And then the other part of this project is using the 2-photon microscope to look at these same brain regions, but the 2-photon will allow us to really look with a higher level of detail at what is going on. So we're kind of just starting this work at the moment, but this is kind of the long-term goal for this project. Okay, so now to summarise. Hopefully I have shown you and told you a bit about how studying brain activity and how it's such an important way to actually understand brain function in health and in disease. And in New Zealand, we actually do have the capability to bypass expensive commercial systems because we have the capability and skills within New Zealand to actually build our own microscopes. And there's many different diseases which can be modelled in rodents, and these can really give us massive insights into what's actually going on in the brain and to really understand how cells are affected by these different genetic changes. And we hope that in time this will also reveal different treatment avenues for these sometimes horrible disorders. So just to go over kind of what I've told you about the projects that are going on in the lab right now, we are trying to understand better what's going on in autism spectrum disorder. So we're trying to understand differences in sound processing in the cortex. And we're also using Miniscopes to really examine brain cell activity during active behaviour and social interaction. In Alzheimer's disease, we're using the Miniscopes to actually look at brain cell activity during memory tests and try and understand, you know, when animals have impaired memory, what's really going on with their cells or what are they not doing. And in spinal cord injury, we're also looking at cellular activity in the cortex and in the spinal cord and trying to understand what is really changing in terms of the circuits in injury and in treatment. And I just also really want to thank the team that I'm working with. So this is the Montgomery Lab. So Joanna Montgomery is shown in the centre here. She's been my long-term mentor. So I did my PhD with her. And when I returned from overseas, I worked with her again. So big thanks to her and all the rest of the members of the lab. And of course, thanks so much to all the funding bodies that have supported me so far with my research, especially the Neurological Foundation. And yeah, thanks so much for turning up tonight and listening. It's been a real pleasure to share my work with you, and I'd be happy to answer any questions.